your curriculum that will be covered here, 69.1, 2 and 3, where we'll be discussing about the elastic muscular arteries, their ultrastructures and the capillaries along with their correlation with the function. So as I said, arteries are the vessels that are taking the blood away from the heart. So there are large size artery, medium size artery, small size artery, and after that there are capillaries which will finally form the venules and the veins. So this is the classification or the flow chart, how from the heart the blood is taken up. So from the heart it is taken up by the large elastic size arteries. They are really large. The circumference of that hollow is more than 10 millimeter, these large size arteries. After that there are medium size arteries. Like if I talk about large size or elastic arteries, from heart there are the arteries that common carotid artery, brachiocephalic trunk, they are elastic arteries. After that in upper limb you have read about the radial artery, brachial artery, those are muscular arteries. So they are the continuation of these elastic arteries. So these are medium size or muscular arteries which are 2 to 10 millimeter in circumference. Small arteries, after that lower division arterioles will divide into form the capillaries. What is the function of the capillary? Why there is a special blood vessel has been formed in any viscera? In any viscera or localized area, why the capillaries have been formed? So that the exchange of the nutrients can take place through the tissues. So only capillaries can do that, not the elastic arteries. They just have to take up the blood from the heart. Now these capillaries, they go into the post-capillary veins, which are called as venules. Post-capillary venule, then muscular venule. These are also very small sized veins, small vein, medium sized vein and the large vein. Example of any large vein: vena cava, superior vena cava, inferior vena cava. This is how the circumference changes. Now endothelium, as you all told me, it is a simple squamous epithelium which is specialized in the blood vessels and is called as endothelium. Another name for simple squamous epithelium is pavement epithelium. Why it is so called? Because it is a flattened epithelium. Whenever you see the squamous epithelium, it is a flattened one. Flattened nucleus and the flattened cytoplasm will be seen. So it is the inner lining of the blood vessel which is endothelial cells. So these flat cells which we can see inside the blood vessel, this is the squamous epithelium. And if we see the cross section, you can see just a line of the nucleus. Now, the basic structure of blood vessel in histology, we will be reading in 3 layers: tunica intima, tunica media and tunica adventitia. What is the term tunica means? Covering. So 3 coverings are there from inner to outer. Innermost is intima, then media and then adventitia. Then they have been further classified. Tunica intima, first one is the endothelium. The first layer in the tunica intima would be endothelium, which is lined by the simple squamous epithelium in every blood vessel, whether it can be capillary, vein or artery. Then you have some connective tissue underlying that epithelium. Underlying the epithelium is the basal lamina. After that we will have a connective tissue so that the tissues can bind them to the adventitia, to the media. So that you have subendothelial connective tissue, which is a loose type of connective tissue. What does that loose type of connective tissue contains? It has fibers, it has blood vessels, it has lymphatics, so as to provide the nutrition to the epithelium. So this is subendothelial connective tissue, which is comprised of loose connective tissue. Then is the internal elastic lamina. So this internal elastic lamina is forming a boundary of tunica intima. After this internal elastic lamina, which is consisting of elastic fibers, you have tunica media. So tunica intima has 3 layers, subdivisions. Tunica media keeps on differentiating in different type of arteries and veins. There can be elastic fibers, there can be muscular fibers, depending on which type of blood vessel it is. Like if it is a muscular artery, there can be more of muscle cells. If there is elastic artery, then more of elastic fibers will be there. So it is consisting of smooth muscle fibers, elastic fibers, collagen. Then you have fenestrated elastic sheets in the tunica media after the muscle layer or the elastic fiber layer. And then you have external elastic lamina. So internal elastic lamina is a part of tunica intima, and external elastic lamina is a part of tunica media. Then you have tunica adventitia, that is the outermost covering. So outermost covering is always fibrous in nature. So here it is consisting of fibroblast, which has collagen fibers, elastin fibers. So if you can understand these three subdivisions of blood vessel, then you can easily identify these slides of blood vessels, artery, vein and capillaries. And you should know about all these layers, because in the viva and in the diagram you have to make these slides. So for this picture, if I see a blood vessel cross section layer by layer, so this much layer is the tunica intima, that is starting from the endothelium, which is resting on the basal lamina. Underneath this we can see the subendothelial connective tissue, and underlying this layer you can see the line, just a line. This is the internal elastic lamina. Now underneath this layer is the tunica media, which is consisting of the elastic or muscular fibers. This whole is the tunica media. Then you have external elastic lamina. This one is the external elastic lamina, which is differentiating it from the tunica adventitia. You can see this tunica adventitia has been marked in white color so as to show you the fibrous origin of the tunica adventitia. So this picture should be clear to everyone about these three layers. Any confusion in these three layers? You will remember this flowchart. This is really very important part of this blood vessel. Now coming to the classification of blood vessels. You all know it is arteries, capillaries and veins. And arteries we have discussed. It is large size artery, medium size artery, small size artery and arterioles. Capillaries are type of continuous type, fenestrated types and sinusoidal. Then veins, you have small venules, small veins, medium veins and the large veins. Now first of all, coming to the large size arteries, which are elastic arteries. The example you all know is one big example is aorta, whether it is arch of aorta, ascending aorta, descending aorta, abdominal aorta. So all come under this elastic artery. Why does it contain elastic fibers? Why not muscular fibers? Any special reason for these large size vessels to have these elastic fibers? Non-collapsible vessels तो होते ही हैं arteries. Elastic recoil of the arteries can take place. Na? There is a large pressure from the heart that is coming to these arteries. So they are also known as diastolic pump. They will throw the blood from the heart to the periphery. Now, what is the differentiating point between the elastic artery in tunica intima, media and adventitia? It will contain all these three layers, but the internal elastic lamina will not be distinguishable from the tunica media. Why? Because tunica media is containing the elastic fibers, more of elastic fibers. I am not saying it is completely filled of elastic fibers, but it is predominantly having the elastic fibers and some of the smooth muscle fibers. So you have endothelium, subendothelial connective tissue and internal elastic lamina, which is mixed with the tunica media. We cannot differentiate it properly. So it has the layer of elastic lamellae and the smooth muscle fibers. Adventitia is comparatively thin compared to the tunica media of the artery. It is not that much broad as in case of veins. So it is relatively thin. Now this is the histological picture of the elastic artery. So starting from inside, this layer is tunica intima. Innermost one will be the endothelium. This layer, first layer that come in your eye contact will be the endothelium. Underneath this tunica, this endothelium, we will find the subendothelial connective tissue. Now we can see this tunica media, they are all comprised of these elastic fibers. Now can you differentiate internal elastic lamina here? No, just we can see that this is the beginning. After the loose connective tissue, there are elastic fibers which are lying, which are mixed with the tunica media. Then you have external elastic lamina, and this one is the tunica externa or adventitia. This picture will be seen when we show you the slides. This is H&E stain, and can you differentiate any of the layers? No. So when you will see the slide, it would look like this only, because this is a handmade diagram where we have differentiated everything, and this is at the 10x. So 10x magnification doesn't give you a bigger picture, it is a very small picture where you can just see a little epithelium. Inside one, you have to assume that it is an epithelium here, where just you can see dotted lines, and underneath these all three layers are mixed up. So little one underlying this endothelium is the subendothelial connective tissue, and underneath that this whole is the tunica media. So what we can do is we can just compare this length of tunica media. This is large compared to the adventitia as well as to the intima. This is a staining for the elastic fibers. If we want to differentiate that this type of any of the tissue is containing more of elastic fibers or muscle fibers, we do a staining which is called as Verhoeven von Giesen stain, which stains the elastic fibers separately. That you can see these are black lines here, dark brown black lines. These are all elastic fibers. So we can now say that this is predominantly elastic fibers in the tunica media. Next is the muscular artery or the medium-sized artery. Another name for the medium-sized artery or muscular artery is, these are conducting vessels. Now in this medium-sized artery, which are distributing vessels, this tunica intima consists of endothelium, scanty subendothelial connective tissue, and now we can differentiate the internal elastic lamina because the middle layer is comprised of the muscle fibers. So more of the smooth muscle fibers will be there. Why not skeletal muscle fibers? Skeletal भी हो सकते थे, but यहाँ पे smooth है. Voluntary fibers होते हैं क्या arteries में? Involuntary होते हैं. तो smooth muscle fibers होंगे. So smooth muscles are present in the tunica media and external elastic lamina will be there. Then you have tunica adventitia. Now the tunica adventitia is thicker than the elastic arteries. Elastic arteries में थोड़ा सा था बहुत. यहाँ पे थोड़ा thick है. So this is the handmade diagram. This is endothelium with subendothelial connective tissue. Now we can see the elastic fibers here. So internal elastic lamina is there. Then you have the tunica media, which is consisting of the muscular fibers, and this is this whole. This is the adventitia. Now you can compare the media and adventitia. Adventitia is comparatively more, as was in the elastic arteries. This is a comparative picture to the vein. So this is an artery and this is a vein. If I take the cross section of this or higher magnification, we can see the wavy pattern in the artery. So this is the endothelium, and this wavy pattern that is seen here is the internal elastic lamina. These are the muscle fibers, and this is adventitia. Now what we can find in this picture, if we compare the medium-sized artery with the medium-sized vein, what you can see? क्या कम है, क्या ज़्यादा है? Lumen. Never comment on the lumen. While making slide, the artery can also show you the collapsible lumen. Next, tunica media. किसका thick है? Artery का. Adventitia? Vein का. So now this is the difference between elastic artery and the muscular artery. Go through it once. So you may be asked this question, list the difference between the elastic artery and the muscular artery. So talking about intima, endothelium दोनों में होगा. सब एंडोथीलियल टिश्यू में कुछ डिफरेंस होगा. सब एंडोथीलियल टिश्यू किस में ज्यादा होता है? इलास्टिक में ज्यादा होता है. देन कमिंग टू इंटरनल इलास्टिक लेमाइना, इलास्टिक में इनडिस्टिंग्विशेबल होता है. मीडियम साइज आर्टरी, इट इज डिस्टिंग्विशेबल. Medium very clear that it has elastic fibers, and in muscular artery it has smooth muscle fibers. Adventitia, less of adventitia, and this is having the more adventitia. The muscular arteries, they have thicker adventitia compared to the elastic arteries. Examples you all know, the arteries near to the heart, the elastic arteries, arteries away from the heart, starting in the upper limb from the axillary artery, brachial artery, those all are muscular arteries. Now what are capillaries? That was all about the arteries. Now what do you understand by a capillary? Capillary is also a type of blood vessel, but there is a lot of networking in the capillaries because there are very smaller branches that keep on dividing, and then they exchange the blood, oxygen, nutrients to the tissues. What is the size of a capillary? It is in micrometer. Those who are in the millimeter, these are micrometer in size, 8 micrometer. ठीक है? So range is from 4 to 10 micrometer. The capillaries micrometer में होती हैं, arteries, veins millimeter में होती हैं. इतना भी याद रखोगे, that would be enough. Now the peculiarity about these capillary is no tunica media and no tunica adventitia would be there. So we will be discussing about the tunica intima only. Now three types of capillaries are there: continuous capillary, fenestrated capillaries, and sinusoids. Now what is the difference? As the name is suggesting, continuous, that means there will be no break. And fenestrated का मतलब क्या होता है? There are pores are present. And discontinuous, that is the sinusoids, they have more breaks compared to the fenestrated. Continuous में क्या है? Uninterrupted endothelium. कोई endothelium में break नहीं है. Continuous endothelium है, एक दूसरे से लगा हुआ. Tight junctions हैं, that is the junctions of the epithelium that are present at the sides of the epithelium. So all the tight junctions are present in case of continuous capillaries. And the basal lamina over which the endothelium is lying, that is also continuous. So कोई break नहीं है capillaries में. In fenestrated capillaries, you have fenestrated endothelium, but the basal lamina is continuous. In sinusoids, both the endothelium and the basal lamina, they are having the breaks. Now I'll show you the picture. So here in this picture, first one you can see there is continuous epithelium is there. This endothelium is continuous, कोई break नहीं दिख रहा है. And if you compare it with the fenestration, here you can see the breaks in the cross section. These are the breaks, and here this is the fenestration. So endothelium में fenestration है, तो वो fenestrated epithelium है, but you have to differentiate it the sinusoid. Now the basal lamina in case of the fenestrated capillaries is intact. इसमें कोई fenestration नहीं होगा. But here is the basal lamina will be fenestrated. So that is the difference between the continuous, fenestrated and the sinusoids. Continuous में क्या होगा? Continuous is as continuous endothelium, continuous basal lamina. Fenestrated में क्या होगा? क्या होगा? Cannot hear you. Fenestrated है तो क्या होगा? There is a break between the endothelium. Pores are present. Basal lamina is intact. And the third one, sinusoids, both are fenestrated. Endothelium and the basal lamina. This is the cross section where we can see the continuous capillaries. So you can also appreciate this basal lamina is continuous, endothelium is continuous. Here we can see the fenestration in the endothelium, but the basal lamina is intact. That means it is a fenestrated capillary. While in the third one, we can see there are lot of breaks in the endothelium as well as the basal lamina. So now coming to the veins, the last part of this lecture. veins. What does the vein do? They carry all the blood from the capillaries to the heart. So how these veins will take up the blood? Through the venules. Post capillary venules will be there, and the muscular venules will be there, which will join to form the small veins, and they will join to form the medium size vein and the large size vein. Now the medium size veins, what is the size is 1 to 10 millimeter. Differentiating point between the intima, media and adventitia. Adventitia is thick compared to the arteries. It is really thick, having the fibers would be same, but the thickness will be different. So fibers are again collagen fibers and elastic fibers in the adventitia. Media is having the smooth muscle fibers, but the media is very small. जैसे elastic muscular arteries में देखा था, there was a lot of muscle fibers are there, but in veins you will find little much of the media. And in the intima, we have endothelium, basal lamina, subendothelial connective tissue, and internal elastic lamina would be there. So this is a picture of the medium sized vein, where we can appreciate the endothelium, subendothelial connective tissue, a little of tunica media is there, and lot of adventitia is present. So how we identify this slide? By the less of tunica media and more of tunica adventitia. Now in case of large veins, what is lesser in intima? We cannot find the internal elastic lamina. In tunica media, now read this line, it is relatively thinner than adventitia. Adventitia of vein. Here it is talking about it is thinner compared to the tunica adventitia. So this point is clear that adventitia in case of veins is always more compared to the media. So you will look for the thickness of the adventitia. So here is the handmade diagram for the large size vein, where you can see lot of adventitia is there. This whole length is showing you the adventitia, and this is the tunica media, and little one is the tunica intima. Now what are the difference between arteries and veins? This is another question that is asked to you. So till now we have read about arteries, capillaries and veins. So if we talk about the wall of arteries, आपने टच करके भी देखा है ग्रॉस में, how the wall of arteries feel like they are thicker. Veins कैसी होती हैं? बहुत thin होती हैं. Grossly देखा है. Now we have to appreciate it histologically. Lumen. The lumen of the artery in gross you have seen it is patent. And here it is not well defined, it is collapsible lumen. Internal elastic lamina in case of arteries is well defined, while in case of vein it is not well defined and mostly it is absent. Internal elastic lamina is absent. In tunica media and adventitia of the artery and vein we have compared. So media, it has thicker than veins. If we compare the media of this artery with the veins, the media will be thicker. Media हमने vein में देखा बहुत कम था, less of smooth muscle fibers थे. But in case of arteries, we have seen lot of elastic fibers in elastic arteries, lot of smooth muscle fibers in the medium size arteries. Adventitia was more thicker in veins. So all the layers of this artery, tunica intima, media and adventitia were well defined, but here it is not clearly identifiable. आर्टरी में इंटरनल इलास्टिक लेमाइना भी पता चल रहा था, एक्सटर्नल इलास्टिक लेमाइना भी पता चल रहा था, बट इन केस ऑफ वेन्स इट वाज indistinguishable. So very less of fibers and less of distinction is there in the veins compared to the arteries. Okay, how do you think would the histological picture would look like if you see elastic fibers and if you have to differentiate it with the smooth muscle fibers? Identify कैसे करोगे? Muscles have been discussed in histology with you, and fibers has also been discussed, connective tissue. Yes? Don't give that blank look. Histology में हो चुका है ना? हाँ. So how do you see those kind of elastic fibers? What is the histological picture of an elastic fiber and a smooth muscle fiber? कितने टाइप के फाइबर्स होते हैं? 3 टाइप के फाइबर्स होते हैं, कौन कौन से? थर्ड लास्ट रो आप बताओ। नहीं आप नहीं, पीछे लास्ट रो। यस, हाँ जो पीछे देख रहा है। यस, योर नेम? हम्म, क्या नाम है? कुलदीप। हाँ बताओ फाइबर्स। फाइबर्स क्या होते हैं पता है? कहाँ से आते हैं? नेक्स्ट, वी हैव डिस्कस्ड फाइबर्स। I'll make easy for you. We have discussed it in connective tissue. अब कुछ याद आया? कनेक्टिव टिश्यू में कितने टाइप के फाइबर्स पड़े थे? 3 टाइप के फाइबर्स थे, कौन कौन से? कोलेजन फाइबर्स और चलो 2 बता दिए तुमने। नेक्स्ट, बैठ जाओ तुम। तुम नहीं। हाँ, नेक्स्ट, कैसे आइडेंटिफाई करेंगे इलास्टिक फाइबर्स? ओके, okay, what type of fibers we can see here in the tunica media? यस सर, आपसे ही पूछ रही हूँ, इधर उधर मत देखो, पता है तुम्हें, खड़े हो जाओ। इलास्टिक फाइबर्स हैं, इतनी देर से बोल बोल के थक गई। मीडियम में स्मूथ मसल फाइबर्स होते हैं, एक यही चीज याद रखनी है और वही याद नहीं है। नेक्स्ट, कैसे आइडेंटिफाई करेंगे? The take home message from this lecture is you have to remember these 3 layers, and you have to remember that elastic arteries, they are having elastic fibers in media, muscular arteries are having smooth muscle fibers in medium size arteries, and all the layers in arteries, they are well distinguishable compared to the veins. This is what would be asked at the UG level. Next, next, खड़े हो जाओ, खड़े होते जाओ जहां तक नहीं आता है। नेक्स्ट रो, मसल्स पढ़े थे? स्केलेटल मसल, स्मूथ मसल, कार्डियक मसल पढ़े थे? 12th में पढ़े थे? तो यहाँ हिस्टोलॉजी के लेक्चर में क्या कर रहे थे? नेक्स्ट, नेक्स्ट, आता है किसी को? नहीं आता? सब खड़े हो जाओ। तुम्हें भी नहीं आता? नेक्स्ट रो, question is how you will identify the elastic fibers and the smooth muscle fibers. Try. Yes, you can try. No, it wasn't. It is not a web-like. Smooth muscle is right, that it is a spindle-shaped fiber with a nucleus at the center. This is how you differentiate, and how this is a characteristic feature of the smooth muscle fiber. But how you identify the elastic fibers? The branching fibers are reticular fibers. No, they are not bundled. Collagen fibers are bundled. They run parallel to each other, and the bundle branching is there in the collagen fibers. ये दिवाली ब्रेक का हाल है. तुम लोग बताओ. नहीं, लास्ट रो. I've completed the lecture. Now I'll be asking you the question only. किसी को नहीं आता? They are singly present like strands. A bit closer. बैठ जाओ. Next. Next row. स्केलेटल को अगर हमें दिस इलास्टिक फाइबर्स को कोलेजन से डिफरेंशिएट करना पड़ गया तो कैसे करोगे? अगर कोलेजन फाइबर्स बंडल में रन कर रहे हैं, पैरेलल हैं एक दूसरे के फाइबर्स, बंडल ब्रांचिंग हो रही है, तो इलास्टिक फाइबर्स कैसे होते हैं? कुछ याद नहीं आ रहा? वो तो स्पेशल स्टेनिंग में होते हैं ना. सिंगल सिंगल कैसे होते हैं, वही तो पूछ रही हूँ मैं. छोटे होते हैं, बड़े होते हैं, लॉन्ग होते हैं इलास्टिक फाइबर्स? नहीं. वेवी होते हैं. हु सेड? वेरी गुड. So elastic fibers are wavy. They are short fibers. बैठ जाओ. ये इलास्टिक फाइबर्स का पिक्चर दिखाया था. These are short wavy fibers. But no branching is there. रेटिकुलर फाइबर की पिक्चर होगी तो कंप्लीट ब्रांचिंग होगी, पूरा नेटवर्क होगा. जैसे जाली होती है, दैट इज ऑल रेटिकुलर फाइबर. और रेटिकुलर फाइबर स्पेशलाइज्ड टिश्यू में ही मिलता है. किसमें मिलता है रेटिकुलर फाइबर? लिम्फॉइड टिश्यू में. So anything you want to ask? अगर इस लेक्चर से इंटिमा, मीडिया और एडवेंटिशिया भी याद नहीं हुआ ना तो वेस्ट है. बिकॉज हिस्टोलॉजिकल स्लाइड्स आपको दी जाएंगी एग्जाम में. यू हैव टू आइडेंटिफाई द बेसिक क्वेश्चन. इंटिमा, मीडिया और एडवेंटिशिया भी नहीं बताओगे, नहीं बनाओगे डायग्राम, तो क्या मार्क्स मिलेंगे तुमको? So प्लीज स्टार्ट रीडिंग. तुम लोग तो सेमेस्टर एग्जाम भी आ गए. यस, आफ्टर लोअर लिम्ब तुम्हारा एग्जाम है ना. सो यू हैव ऑल द सिलेबस: जनरल एनाटमी, हिस्टोलॉजी, एम्ब्रियोलॉजी, अपर लिम्ब, लोअर लिम्ब.